everybody who's joining online, although this is not the most ideal way of interacting, but it also is more time efficient, especially since many of you are in different geographic locations. So also I would like to thank the organizers, especially Professor Proshun Trivedi, for inviting me. And I look forward to a very interactive session with you over the next hour or so. Before I begin, I would like to say that although I'm not in a dedicated teaching institute per se, but I love to teach and I love to interact with students. So anytime during my presentation, if you need any clarification, please feel free to raise your hand and I'll ask the moderator to so that we can take questions during the talk itself. As for the talk, I have tried to keep data to a minimum so that I do not want to take you into a deep dive of data. My goal today is to show you how we can engineer yeast strains to do things in biotechnology. Okay, so I'm going to begin by sharing. And okay, is my screen visible to everybody?
Yes, ma'am.
Okay, thank you. Okay, so specifically, like I said, I'm going to talk about bioengineering yeast. And this, in our case, in my lab, we do it for actually, part of my the taskbar at the end of the screen, is it also visible to you guys? See where
Yes, ma'am.
the okay, so that means part of the presentation might be sort of occluded by the taskbar, but I don't know how to get around it. So let's hope that it's not too much of a disruption. Okay, so like I said, our goal, part of my lab studies yeast. So as Payal already said, I'm a cell biologist and I am also a geneticist. So my PhD and postdoc is in yeast genetics and using yeast genetics to understand various cell biological processes. So I have experience in studying nuclear cytoplasmic exchange as well as proteins being trafficked to the yeast vacuole. After my lab was established, I also study an enteric pathogen called Giardia lamblia, but today my talk is exclusively on yeast. So right now, what my lab aims to do is bioengineer yeast so that they can tolerate higher amounts of ethanol. So yeast and ethanol are very tightly linked to each other because this is one of the oldest sort of biotechnology applications that humans have been doing, that is using yeast to ferment sugars to generate alcohol. So alcohol usage has been documented since prehistoric times, and even pots from ceramic vessels and pots from Egyptian archaeological sites show that they were using yeast to produce wine and beer. Okay, so the connection between yeast and ethanol, as I've already said, goes back a long, long way. But we are increasingly becoming aware of the use of ethanol, not just as a beverage, but also as a means of addressing our energy security. What do I mean by that? You probably know about bioethanol blending in petrol and other fossil fuels, and I'll talk about that in details a little later on. So bioethanol has, or ethanol produced by yeast, has many, many applications. So besides there being a huge market for consumption of ethanol as a beverage. Ethanol is also used in industry as a solvent. And like I said, ethanol is now being used as uh, uh, for blending into fossil fuels. So I wanted to start off by not just talking about ethanol, but biofuels in general. So what are biofuels? So fuel, as you know, is a very, when we talk about fuels, what comes to our mind is um, petroleum products such as petrol and diesel. You can think about LPG. Essentially, all of these fuels are very highly reduced carbon that can be rapidly oxidized, and during that process of oxidization, heat is released, and we harvest that heat and use it for other purposes. Okay, so whether it's burning fossil fuels in an internal combustion engine to drive our transport, or whether it is igniting uh, LPG for cooking, all of this are fuels that modern society is totally reliant upon. So we cannot think of life without using some kind of fuel or another for almost every instance of our daily life. So now the harmful effects of fossil fuel are known to you all. And I'm not going to go into a lot of details about it, but you know about the fossil fuels impact our environment. Fossil fuels are causing climate change. Fossil fuels are also hazardous to our health. So there is now the age of the green economy that's dawning, and this is a very exciting time because of all the research that is going in, and we are getting a lot of results. It's very clear from the number of electric vehicles that you see on the roads that fossil fuel usage has decreased quite a lot, but we have a long way to go. So of these fossil fuels, besides bioethanol, there's also biomethanol, there's biobutanol, there's some forms of biodiesel, and even bio-oils. So all of these are, the term bio means that they are produced from uh, biological organisms. For that matter, fossil fuels were also biological organisms which have been fossilized. But when we talk about biofuels, it is getting fuels out of living organisms. So using living organisms to produce fuels. So bioethanol, for example, it is the raw material. So when we talk about living material, what is raw biomaterial that is used for bioethanol production? So mostly these are either monosaccharides, so these are carbon sources. So it could be sugars, it could be polysaccharides. When I say sugars, it could be monosaccharides like glucose, or it could be molasses. So these are carbons that are being carbon compounds, carbon sources that are being harvested from living organisms. Okay, and for so these are when we talk about energy crops, crops that have a lot of carbon material, that could be sugarcane, that could be beet, that could be sugarcane, sugar beets, and even if you can see this grass may look a little familiar to you. This is a grass that the scientific name is miscanthus, something that we know as kashful, right? So this is also proving to be a very high source of uh, uh, carbon compounds that can be converted into biofuels. So whether it's bioethanol or biomethanol, these all depend on these high energy crops. But you also can get oils like vegetable oil from rapeseed oil, from rape seeds, from sunflower, from soybean, and these are also then converted to fuels. So the conversion processes vary depending on the source. So it could be gasification in case of biomethanol, where the energy crops are converted to methanol. It could involve pressing and to get oil out of seeds. High oil containing seeds are pressed. So whether the oil is used for cooking medium, some oils are not uh, safe enough for human consumption, and those are used for uh, uh, generating biofuels. And also, like I said, in certain cases, the oils are treated differently. So it could be just refining, as in the case of vegetable oils. For biodiesel, this requires a different kind of processing, which means esterification. And then bio oils are produced by pyrolysis, which is essentially taking these high carbon crops and burning them. So when I say burning, it's not just burning, but also certain chemicals are added. So it's a thermochemical process that takes place in the absence of oxygen, and as a result, you get various products such as biochar, which is the solid material. The liquid that comes out is known as bio oil, and you also get some biogas from this. So the whole point of this slide is to make you appreciate that biofuel is not just bioethanol, it could be other things. And the sources of biofuels are high energy crops, and they can even be other agricultural wastes. They could be plant material, sort of wood, dust, and by other byproducts that can be subjected to different chemical processes, and depending on that, you get material that has different applications. For example, bioethanol is used as a gasoline additive or a substituent. You can use biomethanol in fuel cells to generate electricity. And in addition to that, you can get bio oils and vegetable oils, that biodiesel, for substitutes or addition to diesels. So of this, I would like to concentrate on bioethanol. And again, bioethanol is an alcohol. So all of you know the formula is CH3, CH2OH. So there are two carbon atoms, and then it has a lot of hydrogen, only one oxygen, and that's also present as a alcohol group. So consequently, there's a lot of reduced carbon in this molecule, which means that if you burn it, if you oxidize it, the process of converting ethanol to carbon dioxide generates a lot of energy. And so bioethanol, therefore ethanol for that matter, has you know that if we burn it, it burns very cleanly, producing almost all carbon dioxide with very little soot in the form of carbon. So bioethanol is produced, as I've already mentioned, by microbial fermentation of carbohydrates. And again, just to remind you, the carbohydrate source could be from food crops, it could be lignocellulosic material, meaning that these could be wood chips and other other plant that we do not consume. So what are the uses of bioethanol? So as I said, bioethanol usage in energy production, in addressing our energy concerns, are becoming increasingly very, very important, especially in India. So bioethanol has been blended into ethanol, and when bioethanol has been blended into gasoline, and when bioethanol forms instead of burning gasoline entirely in a car internal combustion engine, if you have bioethanol, like I said, it burns cleaner than gasoline, and therefore it is a much better fuel alternative because it produces less pollution, and it is burnt almost completely and converted into carbon dioxide during the process of burning. So you get more heat, therefore more energy out of this bioethanol burning rather than fossil fuels. And at the same time, so the engines become more efficient, but at the same time, you reduce pollution. So bioethanol blending was actually the first country to really understand the utility of bioethanol blending into gasoline was Brazil. The reason is because Brazil has a lot of um, agricultural land which was devoted to corn production, and the corn that was not consumed by humans or not used as cattle feed, to look for a way to use that corn. They understood that corn, if you've eaten corn, which is corn is very has a very high sugar content, because when you bite into it, it tastes very sweet. Sweet corn particularly produces a lot of sugar, so corn has a high sugar content, and therefore corn was used to convert to used as the carbon source for yeast to ferment it into bioethanol. So the rest of the world started catching on, and in India particularly, as you know, we are a net importer of energy. We import almost 70% of our gasoline that we need, the petrol and the diesel that runs our trucks, buses, cars, everything is imported. So that means that we have to spend a lot of our foreign currency reserves towards running our industries and keeping India mobile generally. So, and therefore, not just from an environmental concern point of view, just from also an economic point of view, and in terms of addressing our own energy security, bioethanol blending into gasoline has been the government's focus over the last decade. So if you see, these are numbers that are taken from the Press Information Bureau. In 2014, only 1.53% of the fuel that was uh, being used to power our transport sector contained bioethanol. That blending climbed up to 15% in 2024, and in 2025 our goal is 20% blending. So bioethanol blending into gasoline has taken off in a big way in India, and the government has a huge push in this, our switch from a fossil fuel economy into a bioethanol driven or a bio fuel driven economy. So you can see here the numbers where there has been a almost 15-fold jump over the last decade of the amount of bioethanol that is being used for blending into fuel. So besides, like I keep saying, besides lowering our fossil fuel consumption, besides reducing carbon emissions that contribute to climate change and also to health issues, and also besides enhancing our energy security, making us more self-reliant in terms of the energy that we produce ourselves, there's an added benefit that as a country located in the tropics that we enjoy. So our growing seasons, in whether it's for food crops or energy crops, is basically year round. Most of the country is not plagued by frost. So from January to December, you can produce biomass, and that biomass can then be utilized to address our own energy and food security. And when you shift the focus from just think of this, when India drills for fossil fuel, the economy, that the impact on the local economy is sort of concentrated to the regions where the drilling is taking place, whether it's offshore drilling in Mumbai or it's drilling in Assam. That's where the economy is directly benefited because of these activities, and that is very localized effect. On the other hand, when you produce a biofuel based economy, in a biofuel based economy, the entire country's rural economy stands to gain. So taking all of this into account, this is one of the, it's a very good reason for us to switch our focus from being a fossil fuel driven economy to a biofuel driven economy. So whether you're talking about environmental sustainability, whether you're talking about economic development, or you're talking about energy security, it's a win-win situation when we move to a biofuel based economy. So you can see again here, this is data from the Press Information Bureau site, where we've saved considerable amount in foreign exchange, we've reduced CO2 emission, and we have done a significant amount of crude oil substitution by using biofuels. But this substitution, we need to do more, and other technologies such as car engines being converted to be able to use more higher amount of bioethanol, that needs to be done. So let's now talk about what are the things that go into producing biofuel or bioethanol in particular. So as I've mentioned before, that the first country that really took up bioethanol blending into fuel in a big way was Brazil, and Brazil did that because they had a surplus of corn. So when it comes to these the biomass that is used to generate biofuels, we've also undergone a transformation. So the first generation biomasses that were biomass input that was used was again food crops, corn, sugarcane, wheat, beet. These are all why were they used? Because they're very highly enriched in sugars. So these serve very quickly, you can get bioethanol from these sources. But we'll see that there was a challenge, because it's like choosing whether you put food on the plate and or you convert that food into biofuel. So it's, you know, that food security is also a very important parameter for the world, and there's a lot of food shortage. So it's not as if we have excess food grains that we can now use to convert to bioethanol. So the hunt was on for using other forms of carbon. So the second generation of these feedstocks, where technology was developed to use non-edible biomass, such as wood, whether it's wood that is grown from social forestry or wood products, byproducts of wood being used in industry. So wood is now being used. You have straw. So all of you know that there's this huge problem of crop residues in the country, and it contributes to pollution, especially in our national capital. So the straw, the leftover from agricultural activity that can be used for biofuel production. Grass that grows, so I talked about kashful. So this is again grass that is not edible but grows wild, that can be used. And then there is also waste. So we are talking about the circular economy, where waste that is generated because of human activity can, instead of contributing to environmental pollution, that can be reused, sort of recycled to produce, not recycled, but rather used to produce biofuels. And the third generation is where, again, all of these, whether it's the first generation or the second generation, this requires land, this requires fresh water, because you have to grow. But even if you're not growing sugarcane or corn for biofuel production, if you're going to rely on forests, if you're going to rely on trees for wood or grass, you need land and you need fresh water. And in that way, you are now again, generating these resources competes with resources that are needed for food production. So in the third generation of biomass production, the focus has now shifted to generating biomass that does not require land and does not require fresh water. What is this biomass? How do you grow it? You grow it in the sea, and so you're not using up any land, and the seawater is being used, which we do not use for agriculture. So algal biomass is now the third generation of biomass that is being used for biofuel, whether they are microalgae or macroalgae. There's a huge investment that is research, a lot of R&D activity in algal biomass production, and large companies are looking at this in a very, are putting in a lot of effort. Okay. Now comes the fourth generation of biomass production. So what is the fourth generation? This is where genetic tools come in for biomass production. So let's say you have a biomass, but you want that biomass to now grow more quickly. So you can understand whatever organism or whatever carbon source providing organism that you're growing, whether it's algae or whether it's plants, you bioengineer these so that they grow quickly. So when they grow quickly, they fix atmospheric carbon dioxide, they utilize atmospheric carbon dioxide and convert it into biomass. So during the process of this quick conversion of carbon dioxide, you reduce greenhouse gases, and at the same time, you produce a lot more biomass in a shorter amount of time. So this is the idea behind using genetics tools, to use genetic engineering to create high biomass yield. The other thing is also, when you use plants such as grass and wood, besides the cellulose, which is going to get converted into sugars that will get converted into ethanol, let's say, plant residues also contain non-sugars. These are called lignins. Lignins are the tough materials that help plants stand tall. So when you look at a giant sequoia tree, it is the lignin content in that plant that is allowing it to reach those incredible heights. But this lignin is not suitable for being converted into sugars. So if you can engineer biomass generations where the lignin content is low, you can actually improve the efficiency of biomass being converted to much more sugars. So not only do you want plants to grow quickly, you also want to grow plants that have low lignin content. And if they grow quickly, then they also enhance carbon storage. So if you have plants that store a lot of sugars. They instead of using them, they have the ability to store carbon as sugar, store the sugar. Then again, you are enhancing the biofuel production ability for that plant. So all of this together is, I hope you can understand how there has been a very rapid change in the kinds of biomass that we use. And when we use that why is that rapid change happening? Because we want this biomass to be produced rapidly, but at the same time, we don't want biomass production to compete with food production. So again, to go over that in a little more detail, if you can see over here, first generation biofuels were mainly food crops, whether it's corn, soybean, sugarcane, etc. They were used because it was a simple process. I'm sorry, is there an extra window that's interfering with the presentation, or you don't see it?
Yes, ma'am. There is an extra window unpacking.
Okay, now that's
Yeah,
gone.
now
Yeah.
it's fine. Yeah.
Okay. Sorry, that happens with my computer sometimes. So, okay. So again, getting back, so when you are using food crops, it's a simple process. It's more cost efficient because you have a lot more simple sugars that the plant is producing that can be rapidly converted into bioethanol. And it doesn't require any pre-treatment. You don't need any pyrolysis. But as this slide puts it, there's a food versus fuel conflict. So it's taking away food crops to produce energy rather when we do have starving individuals all over the globe. So that's not an ethical solution that, people in more fortunate people living in developed and developing countries should have, uh, that we reduce pollution for them while there are individuals who are starving. Also, there is the issue of land use and freshwater use. That's why we moved to second generation biofuels, where we are using organic waste, we are using agricultural residues, wood, etc. These biomasses have high lignocellulosic lignin content, and therefore they are not as efficient. The advantage was that now you're using waste, so you're increasing the pace of the circular economy because you're removing waste, and it also bypasses the food versus fuel conflict. But the disadvantage is, again, unlike food crops, it's not as easy to convert these second generation biofuels into second generation biomass into biofuels, and they require extensive pre-treatment. And these pre-treatments, if you take everything into consideration, the environmental hazards that are associated with these pre-treatments, the sort of advantage of using biofuels reduces drastically. So that's why there was, in came the third generation of biofuels, where there is no direct conflict in the terms of agricultural lands being taken over for biofuel production or freshwaters being used. And also these do carbon capture, so carbon dioxide is removed. But this involves higher downstream cost in the sense that the algal farms that need to be set up. So there's a lot, it's a very cost intensive process. So and this brings us to the fourth generation, where we are trying to manipulate these living organisms, whether they are plants, coaxing them to grow more rapidly, engineering them so that there is less lignin, or whether it is engineering those yeast that are going to convert the sugars, the simple sugars into ethanol If we can engineer yeast and make them more efficient, then also we would actually make the process of biofuel production more efficient. So this brings me back to yeast, Saccharomyces cerevisiae. As I was telling you in the beginning of my talk, Saccharomyces cerevisiae is a very old friend of ours. We've used Saccharomyces to produce ethanol for over as long as so our association with yeast is as old as human history. So I wanted to, before I start talking about yeast, the pathway by which yeast generates ethanol, I wanted to tell you a little bit about yeast itself. So one of the reasons yeast is used rather than, let's say, other fungi, because those would also produce, or other fungi also have the ability to break down sugars into ethanol. Yeast is used because it's very rapid to grow. Okay, and also it does not, when it converts sugars to ethanol, it does not produce any toxic compounds. So yeast is safe to use. It grows very rapidly. Most of you probably have worked with E. coli in the lab. Just like you grow E. coli, we can grow yeast also very quickly. Usually it's an overnight culture of E. coli. It will take you between 10 to 12 hours. Yeast grows to similar density in about 18 hours. So you can understand with vis-a-vis or with respect to E. coli that yeast is also unicellular and it grows very rapidly. The reason it grows so rapidly is because it can utilize glucose very efficiently. Okay, so it can yeast's genetic circuitry in glucose and other carbon source utilization has been perfected over eons of evolution, to the extent that just like E. coli, yeast can uh, very quickly take in glucose through or other carbon sources through the various pumps and transporters in its cell membrane, and it can efficiently use its metabolism to convert glucose to uh, ethanol, or yeast is not trying to produce ethanol. Yeast is actually, what it's trying to do is extract energy from glucose, right? So it's glycolysis, and even for that matter, when the pyruvate, the product of glycolysis, gets fed into the mitochondria, conversion from pyruvate to carbon dioxide and getting ATP, that's also done very efficiently by yeast. When yeast does not have access to enough oxygen, unlike us, yeast can just survive by substrate level phosphorylation. So over a long period of time, what happens is, if you grow yeast under anaerobic conditions, then the mitochondria shut down, and instead the pyruvate is converted to ethanol. And I'll show you that in a later slide. So if you compel yeast to grow in the absence of adequate amounts of oxygen, you will actually bump up ethanol production. So it utilizes glucose at a much more rapid rate, but instead of being able to convert that glucose into, burn that glucose completely in the mitochondria to produce CO2 and energy, it now produces ethanol. So here I have a very brief overview of the yeast life cycle. I'm not going to go into details, but just as you know, E. coli divides by binary fission. Yeast also does that. Yeast, instead of binary fission, something comparable happens where part of the cell starts forming this bud, and the bud starts growing in size, and finally there is a division of the bud is called the daughter cell, and it segregates away from the mother. And if you look at yeast under an electron scanning electron microscope, this is how they look like. They're not exactly spherical, they are little oblong. So and what you see over here, these little scars, these are actually the spots from where a daughter cell has segregated away from a mother cell. So those are called bud scars. So essentially yeast multiplies by budding. And that's all I want you to know about the yeast life cycle. I don't want to go into the haploid diploid issue. So yeast also, when coming back to how glucose is converted to ethanol by yeast, if you keep them under anaerobic conditions, less oxygen, glucose gets converted to ethanol. Now, when this happens, why is it that we cannot survive for a prolonged period of time under limiting oxygen conditions, but yeast can? This is because, being unicellular organisms which are out in the environment, they have much more sophisticated stress response pathways than we do. So yeast has an inherent high tolerance to environmental stress. I mean, they've been living millions of years in an open environment, not even millions, billions of years in the open environment. And so their biology is more tuned to handling environmental stress better than we can. And on top of that, because of its rapid ability to grow rapidly, and it has a very simple genome, all of this meant that starting from the early parts of the 20th century, researchers have been using yeast as a model organism to study more complex understand biology in general. So many of you may know that most of the cell division cycle genes were actually discovered in yeast, not just Saccharomyces cerevisiae, but another fission yeast called Schizosaccharomyces pombe. So as a consequence of this sort of yeast being very easy to work with, and it having very a simple genetic architecture, and it's easy to grow, cost effective, very inexpensive to grow, it's been studied very extensively. So all the tools that are needed for genetic engineering, such as good vectors, having ways to delete genes, being able to do all kinds of genetic manipulations, being able to insert new genes into the genome of yeast, these tools have been developed very well over the past almost 80 years now. So as a result, it is very easy to engineer yeast when it comes to enhancing its ability to produce ethanol or surviving under stress conditions as ethanol is being produced. So we can do all of this because we've studied yeast's physiology, its metabolism, and the genetics. All of these have been very intensively studied over the past almost 80 years. So now let's talk about how to bioengineer yeast so that it can produce more ethanol. So in terms of bioengineering yeast, let's first understand how glucose is converted to ethanol. So all of you know that glycolysis results in glucose being converted to 2-pyruvate, and this pyruvate then moves into the, let's see, let me get the laser pointer. Okay, here. I hope the laser pointer is now visible. So this pyruvate can then either get fed into mitochondria to produce more energy. But again, when there is a dearth of oxygen under anaerobic conditions, pyruvate gets converted to acetaldehyde, which is then converted to ethanol, resulting in the reuse of NADH generated by substrate level phosphorylation of ADP to ATP. So this NADH is brought back to Hello. Okay, if there's a question, please feel free to stop me. right? So this conversion of pyruvate to ethanol is alcoholic fermentation. So how do we engineer yeast? So we can either metabolically engineer yeast so that it is more efficient in converting glucose to ethanol by optimizing its metabolic processes, or we can use synthetic biology by which we can introduce new pathways that allow yeast to utilize varied carbon sources. So instead of glucose, if we give it now a different carbon source, a synthetic biology modified yeast should be able to use this new carbon source. So we can introduce completely new pathways by synthetic biology approaches and allow yeast to make ethanol from new carbon sources. Or what we can also do is use these bioengineering approaches to allow yeast to survive better in under the stress conditions that result when ethanol is being produced. So I'll come back to the ethanol stress conditions a little later on, but what are the benefits? Let me quickly touch upon the benefits of bioengineered yeast. So again, like I said, metabolic engineering should be able to give you higher ethanol yields. Not only that, if yeast can survive in the presence of the excess ethanol in the medium, if they can survive better, they will also continue to produce ethanol for a longer period of time. This is a tolerance to environmental stressors. So what this means is, as ethanol is being produced, ethanol itself is a toxic product. So that toxicity, if yeast can handle that better, it will make the yeast survive longer in the presence of ethanol. Not only that, ethanol production also leads to environmental stress such as thermal stress. So yeast that can withstand higher temperatures should be able to keep producing ethanol for a longer period of time. And ethanol also causes lowering of cellular pH. How? I will come to my next slide. Even low pH ability of yeast to withstand low pH in the cytoplasm helps yeast survive longer. So and again, there is also, you can bioengineer yeast so that it has the ability to use a variety of substrates, as I was talking about, by introducing new pathways. So using synthetic biology to create new yeast strains that can use a wider variety of substrate besides just glucose and galactose. So now let's try to briefly understand why is ethanol actually causing stress in the cell. So as I said, for ethanol during the alcoholic fermentation process, glucose is converted to ethanol. Now this ethanol is happening without glucose being shunted into mitochondria. This, as a consequence, less ATP is being produced from glucose, because all the high efficiency ATP production in mitochondria is not happening. So consequently, to survive, yeast need to use glucose at a very rapid rate, because instead of being converted to carbon dioxide, glucose is now being converted to ethanol. So there is a rapid depletion of glucose. So ethanol production, in some sense, is also a nutritional stress, because the major carbon source is becoming limiting. Ethanol production also, as a consequence, ethanol production generates reactive oxygen species. So this acetaldehyde that's being produced, that during the production of ethanol, that can actually contribute to the production of ROS. And so there's ROS generation in the process of ethanol production. Also, if you think about it, unlike water, ethanol is also hydrophobic in nature, much more hydrophobic than water. So our cell membrane, the tightness of the cell membrane, these lipids that come close to each other and the hydrophobic cores of the lipids can interact with each other, and the membrane that is formed is very, what should I say, very, very robust in not allowing even something as small as a proton to leak through. That depends on the fact that the lipids are hydrophobic and the surrounding environment is hydrophilic. If the surrounding environment with ethanol production, the hydrophilicity of the surrounding environment decreases, consequently, the lipids do not interact with each other as strongly. So the hydrophobic force holding two adjacent lipid molecules weakens, and as a result, there is a leakage. The membrane is not as tight anymore, so you get hydrogen, protons being leaked. Consequently, there's a loss in lowering of pH, as I already mentioned in the last slide. So all of this causes stress in the cell. And finally, besides the membrane being destabilized due to higher hydrophobicity of ethanol, proteins are also destabilized due to the hydrophobic nature of ethanol. So what I've shown you is that there is, when yeast is making ethanol, there is a nutritional, cells are facing nutritional stress. Cells are facing thermal stress because, again, there's a lot of glucose being converted to ethanol. There's an enhanced glucose conversion, which causes a lot of bond breakage, is causing energy release. So there's some amount of thermal stress. There is reactive oxygen species being generated. So that is ROS is contributing to the stress. There is a lowering of the pH, which again is a stress. And finally, the proteins themselves are not functioning fully. So in all of this, there's one area which I'm interested in, is the role of the vacuole in mitigating the stresses that result from ethanol production. My goal is to tweak the biology of the vacuole so that it is such yeast cells are able to withstand the stress conditions generated by ethanol far better. So as you know, beer is about 6%, 6 to 8% alcohol. Wine is about 12% alcohol. But if you want beverages that have higher alcoholic content, such as spirits, okay, whether it's whiskey, vodka, etc., these are distilled, meaning that these are the yeast generates ethanol. After a certain amount of ethanol has been produced, the alcoholic fermentation process stops because the yeast can no longer survive. Now, after the yeast have died and the alcohol percentage is like 12, 13%, liquid is now distilled so that the alcohol is distilled out and you produce higher spirits rather than. So the difference between wine and something like spirits is that there's a distillation process involved. And the reason the alcohol content of wine doesn't go beyond a certain percentage is because yeast cannot survive. So my goal is to tweak the biology of the yeast vacuole so that it can allow these yeast to continue fermentation at higher ethanol percentages for a little while longer. So yeast that are able to withstand ethanol stress for a longer period of time will allow ethanol production to be enhanced. So we already know why did I become interested in the vacuole? So the vacuole is already known that when the cellular cytoplasmic pH goes down, there are proton pumps in the vacuole that become active that remove the extra protons from the cytoplasm and puts it in the vacuole so that the lowering of pH does not hamper with the functioning of the proteins in the cytoplasm. But just being a pump for having pumps for removal of protons, the yeast vacuole has a lot of other functions. So for example, yeast cells can remove toxic materials from the cytoplasm into the vacuole. The vacuole serves as a storehouse for nutrients. So whether it's so when certain proteins are targeted into the vacuole, they are broken down, and the amino acids that are produced because of this enzymatic hydrolysis of proteins, those are then transported back into the cytoplasm, where they can take part in newer rounds of protein synthesis. It also has pumps to control the amount of cations and anions in the cytoplasm. So I hope you can understand that the vacuole is instead of just being a degradative organelle, the vacuole is actually playing a very important role in allowing an organelle, the yeast, to withstand different stress conditions. And one of the things that we have uncovered in recent years is that besides these chemical activities of the vacuole, wherein it removes protons, it releases amino acids and serves as a storehouse for amino acids, the physical properties of the vacuole are also very important, in the sense that this is a perfect example to show you that yeast vacuole normally has 2 to 3 lobes. So you can see 1, 2, 3 different compartments of the yeast vacuole, which in this case has been stained with a dye. Okay. If you put yeast cells under hypotonic conditions where lots of water is coming in because of the osmotic imbalance, yeast vacuole swells up. And if you do the math, you can clearly see that when there is a large increase in cell volume and the cytoplasm cannot keep up, to compensate for that cell volume increase, a larger vacuole with a lower surface area to volume ratio will occupy more space, thereby compensating for the enlarging cell very quickly. Conversely, when you put yeast cells in hypertonic medium, where lots of water is flowing out, this 2 to 3 lobes fragment very quickly into this little, you can see tiny vacuolar compartment. And again, just as here it was compensating for the extra volume of the yeast cell because of water inflow, here this compensates for the decrease in cell volume because of water outflow or egress. Even when stresses that do not involve water coming in or going out of the cells, such as thermal stress, you can see yeast cells go from having that 2 to 3 lobes into unilobed yeast vacuoles. And in nutritional stress also, we see a change in the vacuole shape. So besides the functioning of the yeast vacuole in various stress elevating low pH, the shape of the yeast vacuole is also important in vacuole stress response. And today, although I will not go into the factors that control vacuole shape, I will just tell you that vacuole shape in yeast changes from 2 to 3 lobes to single lobed or fragmentation, because the whole vacuole shape is controlled by a balance of fusion between different vacuole compartments, and then a large vacuole undergoing fission to produce smaller compartments. So this vacuole shape biology is being increasingly appreciated in the recent years. So very briefly, I'm going to show you 1 or 2 data slides. What we've seen is that when we subject yeast cells to increasing ethanol concentrations, you can see our lab strains stop growing at 12% and they're barely alive at 10%. If we do a time kinetics of exposing yeast cells to ethanol, what you can see here that in control cells, the cells have 2 to 3 vacuole lobes. Very quickly, those lobes fuse and become a single vacuole compartment. And this is also observed with increasing concentrations of vacuole. So I need to switch back to, okay. So I'm not going to show you, this is just a video that shows that the yeast vacuole shape, how the shape changes. So now we've also seen that in case of mutants, and I'm not, in the interest of time, I see that my 1 hour is almost over. So I'm going to very quickly say that we have yeast mutants that cannot fuse their vacuoles. So you can see over here that in under certain conditions the vacuole fuse, but these mutants are temperature sensitive, meaning that higher temperatures they cannot cause their vacuoles to fuse. And when we have these mutants which cannot fuse their vacuoles and make the vacuole a large single compartment, those cells are more sensitive to ethanol. So this tells us that for yeast cells to survive well in the presence of ethanol, they need to fuse their vacuoles together and make one large vacuole. Now you may ask, why is it that yeast vacuole shape has to change from being fragmented or 2 to 3 compartments to a single large compartment? And without going into all the mathematical reasoning, I will tell you that this has something to do with membrane tension. So here I'm showing you an example of a balloon that is fully inflated as opposed to a balloon that is not fully inflated, sort of partly deflated. All of you know from this practical example that the tension of the balloon's the balloon surface is not as much when you have a partially deflated balloon. What happens is that when you add ethanol, if you think back, I told you that because ethanol is hydrophobic, the membrane is not going to be as tightly packed. The lipids are not as tightly packed. So the membrane becomes more fluid. When the membrane becomes more fluid, it results in reduction of the membrane tension. So when you make the surrounding environment more hydrophobic, the membrane of the vacuole goes from being that of an inflated balloon to a partially deflated balloon. The tension decreases. And this decrease in tension has a consequence in the way that all the transporters and the enzymes and the channels that are located on the vacuole membrane. These proteins, these transmembrane proteins, their structure has been optimized to function under a certain membrane tension. If that tension goes down, then these transmembrane proteins are no longer functional. And hence, this membrane tension is a very important physical parameter that dictates the functioning of proteins. Without showing you the calculation, we've done this, and this is very well established, that if you decrease the surface area to volume ratio, then the tension of that membrane, of the reduced surface area membrane, actually increases. So what we now are proposing is that this rapid change in the shape of the yeast vacuole in response to ethanol is actually a very early response that yeast cells have to maintain the tension of the vacuole membrane. And later on, as the yeast has more time to adjust to this change in the physicochemical properties of ethanol-induced changes in the physicochemical parameters of the cell, it's going to bump up other biosynthetic processes to stabilize the membrane. So this first change in the yeast vacuole shape is a quick way to mitigate that stress. With that, I think I've gone a little over time. I'm going to stop, and I will acknowledge my long-term collaborator, Professor Alok Sheel at Calcutta University's Microbiology Department, technical support that we've received from ours is a very highly intense microbial microscopy lab. So there are a lot of technical support involved, and the past and present members of my group. And the little data that I've shown you today is the work that is done by Pritha Mondol, who is a very talented grad student in the lab, a very dedicated and talented grad student. And with that, I'm going to stop and take questions if you have any.
Thank you, ma'am, for illuminating this vital topic. Today, we have gained a deeper understanding of the intricate process of biomass production. And it is also very clear to us that with continued research, the power of the microbes offer innovative solutions. So thank you, ma'am, once again. If there are any questions from the participants, we would be very happy to take that. Any questions?
All right, Payal, I think there are no questions at this point. So again, thank you. Thank you to the organizers, and thank you for the patient listening. Again, this is not a format where I can see the faces of the audiences, so it kind of becomes very difficult, but we are all getting used to the new normal, so as to say. All right.
Ma'am, thank you. Just I have one question.
Sure.
That is that you have beautifully described how yeast is helping in the production of ethanol. So, in your lab, have you worked with bioengineered yeast, or was it the normal yeast that is available in the lab?
So I've not shown, thank you, Payal, that's a very good question. And I have not shown you all the data. There were just two. When we work with Bose yeast mutant cells, so when we generate mutant, that is also sort of bioengineering. And then what we do is we want to make cells that can quickly enlarge their vacuoles, right? So we are trying to drive the process forward by artificially expressing other yeast genes in the cell, sort of elevating the levels of those yeast genes. Particularly, we are trying to elevate the production of a certain lipid. And when we do that, we are using yeast's own genes. So technically, it's not using any genes other than yeast own. But when we are playing around with the levels of the genes, whether or gene products, whether it is through deletion of the gene or whether it's through ectopically expressing the gene at a higher level, that's considered bioengineering, but it's not at the realm of synthetic biology where we are taking other genes from organisms other than yeast and introducing. So yeah, we are still at the basic level. We are trying to accelerate this process, and we'll see if we can do it with other genes more efficiently.
Thank you, ma'am. Thank you once again for your valuable time. We are really honored to have you with us today.
Oh,
Thank you, ma'am.
the honor is all mine. It was really great opportunity. Thank you. Okay.
Thank you, madam. Thank you so much.
Thank you, Prashun. You're welcome as always. Okay.
Thank you.
Okay, participants, now we will dismiss for lunch. The next session is from 2 p.m. So please join accordingly with the same link provided in the group. Thank you.